Good day, grade 11 learners, and welcome to this video where we will be continuing with our control test preparation for grade 11 for term 1. Today we'll be looking at section B questions taken from a previous control test. So we'll first look at the question 2 that starts with 2.1. This question is based on a scientific investigation, and you should have a scientific investigative question in your control test based on either of the topics that were covered in term 1. Either microorganisms, plant diversity, or animal diversity. So scientists conducted an investigation to determine the effect of antibiotic on bacteria causing pneumonia in children from 2000 to 2006. The procedure for the investigation comprised of the following. The bacteria were cultured in the same type of agar medium in petri dishes of the same size. The petri dishes were divided into two groups. Petri dish A was treated with antibiotic. Petri dish B was not treated with antibiotic. An antibiotic was introduced in year 2000 in Petri dish A. Both Petri dishes were incubated under the same conditions and the bacterial growth was examined. The investigation was repeated a number of years. So as you can see here, the ultimate aim of this investigation is to determine the effect of antibiotic on bacteria causing pneumonia in children from 2000 to 2006. They use the same type of agar medium in petri dishes of the same size. You had petri dish A that was treated with antibiotic, but petri dish B was not treated with antibiotic. The petri dishes were incubated under the same conditions and the bacterial growth was then examined. So the table below shows the results of the investigation. You have your years, you have your growth of bacteria per 100 for petri dish A as well as petri dish B. The first question asks you to state the possible hypothesis for this investigation. So the first possible answer you could have said for 2.1.1, you could have chosen petri dish A, and you could have said petri dish A, which was treated with an antibiotic, will cause a decrease in the growth of bacteria, which causes pneumonia. Or you could have chosen petri dish B, and in petri dish B, that was not treated with antibiotics. So therefore you say petri dish B, which was not treated with an antibiotic, will cause a decrease in the growth of bacteria, which causes pneumonia. Or lastly, you could have said that the use of an antibiotic will not have any effect on bacterial growth. Now remember that whenever you are stating a hypothesis, it's a prediction. So what you predict to happen in the experiment. Therefore you have possible answers. There are three possible answers here. It may not necessarily be one answer only that is going to be correct. The next question, 2.1.2, says identify the independent variable. Now remember that when you are stating the independent variable, you need to take it from the aim of the investigation, and this is the aim of the investigation. So the effect of antibiotic on bacteria causing pneumonia. So what is the independent variable? What's the variable that you are changing in the experiment? You cannot use the phrase effect of antibiotic. You need to say something about the antibiotic. So we know in petri dish A, the antibiotic is present, but in petri dish B, the antibiotic is absent. So therefore we can say the content of the petri dishes, so specifically the presence or the absence of the antibiotic, or you could also say the time period, which is going to be in years. Then B speaks about the dependent variable. The dependent variable is the one that you are measuring. So what are you actually measuring here? Causing pneumonia, bacteria that causes pneumonia. So therefore we're talking about the growth of bacteria. 2.1.3 asks us to calculate the average bacterial growth rate per 100,000 in petri dish B with 3 marks. So remember, you need to show your calculations. So we are looking at all the contents in petri dish B. Remember that when you calculate the average, you are adding all these values together. And when you divide it by the number of values that there are. So if you count 1, 2, 3, 4, 5, 6, 7, 8. So you add everything and you divide it by 8. Therefore you have 23 plus 28 plus 30 plus 40 plus 45 plus 50, 55 and 60 divided by 8. Type in the calculator. Gives you 41,38. And the unit of measurement is bacteria per 100,000. Because here we have the unit of measurement in the table. Please ensure that you insert the unit of measurement next to your answer as it contributes to this mark allocation out of 3. So you get 1 mark for the calculation, 1 mark for the correct answer, and 1 mark for the unit of measurement that is correct. The next question, 2.1.4, asks us to describe the growth of bacteria from 2001 to 2006 in Petri dish A. So if you look at Petri dish A, you will see that as the years are increasing, the growth rate of bacteria is decreasing. 2.1.5 says state two factors that were kept constant during the investigation. So in the first slide, we highlighted a few things and we said that the type of agar medium was kept constant, the size of the Petri dishes, the conditions where Petri dishes were incubated. These were some of the things that were kept constant in the investigation. 2.1.6 asks us to explain why Petri dish B was included in the investigation. Now we see that in Petri dish B, that was one where they didn't treat it with antibiotic. Now why could they possibly have inserted one where they didn't treat it with antibiotic? Simply because they wanted to have something to compare the results of the investigation with. But to serve as a control, so it can be compared with group A or Petri dish A. Remember that controls are simply there to serve as a purpose, to have a means to compare your results of the investigation to. 2.1.7 says suggest one way in which scientists can improve the reliability of the investigation. Now remember that there are 5 things that you can do to improve reliability. It's repeat the investigation, increase the sample size, calculate the average of the readings, increase the time period of the investigation, as well as select a random sample. So any one of that would have been correct. The easiest one we could have stated was repeat the investigation. 2.1.8 says draw 2 line graphs on the same set of axes that shows the relationship between the growth of bacteria per 100,000 from 1999 to 2006 in Petri dishes A as well as B. So 2 line graphs on the same set of axes will simply mean that we need to include a key onto this graph. Firstly, make sure that you have the correct heading for the graph. So you state which type of graph you are using, the line graph, and say what is happening in the line graph. Include both variables. So if we look at the answer, the heading line graph showing the relationship between the growth of bacteria, because that's the dependent variable, per 100,000 from 1999 to 2006, because the time period is going to be the independent in Petri dishes A and B. We make sure that the x-axis has the independent variable, that's our time in years. The growth of bacteria per 100,000 is the dependent variable. We make sure we have a correct scale here on the y-axis. On the x-axis, you insert all your time periods in years, so you state all the years. You plot your points and you have an accurate line for Petri dish A, and you have an accurate line for Petri dish B. And then you plot your points of Petri dish A and you join the dots to draw your line graph. You make sure that you have a key here at the side of your graph because you are drawing 2 line graphs on the same set of axes. Just to go through the criteria that we will use when marking the graph, 1 mark for the heading, 1 mark for the correct type of graph, so if it's a line graph, 1 mark if you labeled your x and y axis correctly. Remember that if you transpose the x and y axes, the variables on the x and y axis, you will lose a mark. 1 mark for a correct scale, 2 marks for plotting your points correctly. The total mark allocation comes to 6 marks. The next question, 2.2, says draw a label diagram that shows the structure of a bacterium for 5 marks. Please ensure you are able to draw and label the different types of microorganisms that were studied, namely the virus, the typical bacterium, amoeba species for protists, as well as bread mold, also known as Rhizopus species. In addition to that, in animal diversity, you also need to be able to know how to draw and label the body plan of a diploblastic as well as a triploblastic organism. To answer this question, the structure of a typical bacterium, this is the structure of a typical bacterium. Please ensure that you state the label of your drawing at the bottom of the drawing and you have at least 3 correct labels for your drawing. For example, cytoplasm, flagellum, nucleoid, cell wall, slime capsule. The criteria for marking this drawing is, if it's out of 5 marks, you'll have 1 mark for the heading, 1 mark for the correct drawing, so because you drew the bacterium according to what the question instructed you, 1 mark for that, and any 3 correct labels Gives you total mark allocation out of 5. Please ensure that you comply to this criteria so that you are guaranteed that you get that full 5 marks for 2.2. The next question, question 3, says complete the following table showing different animal groups with their corresponding features and write down only the number of the question and the answer. So for example, 3.1.1 and your answer, 3.1.2 and your answer, which will be stated in your answer booklet. So let's look at what is actually happening here in the table. Firstly, you have the animal group, the phyla, the named example, type of symmetry, tissue layers, and lastly, gut openings. Just a reminder that when you are studying your animal phyla, if your textbook has a table that summarizes all this information, please use that table to summarize it and study the information from the table, because it will really assist you in actually understanding all the characteristics of the different animal phyla that we studied in animal diversity. The 3.1.1, the animal group, the name of the phyla that's referred to as sponges is going to be Porifera. 3.1.2, the number of tissue layers in one that has radial symmetry, that's a jellyfish, that's Cnidaria. How many tissue layers are there? Now we know that Cnidaria is going to be diploblastic, therefore it has the ectoderm and endoderm. So we say diploblastic, comprising of the ectoderm and endoderm. The next question, 3.1.3, asks us to differentiate between diploblastic and triploblastic animals for 2 marks. So what do we mean by diploblastic and what do we mean by triploblastic? Diploblastic are animals that produce only 2 germ layers, that's namely the ectoderm and endoderm, but triploblastic animals produce 3 germ layers, comprising of the ectoderm, endoderm, as well as the mesoderm. 3.1.4 says state one disadvantage of having a blind gut in an organism mentioned in question 3.1.2. So what does the one disadvantage of a blind gut mean? B. Remember that in a blind gut you only have 1 opening, but in a through gut you have 2 openings. So if you're only going to have 1 opening, the animal cannot ingest and egest at the same time. The ingestion, digestion and egestion are not systematic. So you can't have all these processes occurring at the same time, because you need to ingest and ingest food from the same opening. So that is going to be any one of these disadvantages would have been acceptable. You could also have said there's mixing of digested and undigested food. Why? Because it's all coming in and out of the same opening. (3.1.5) says explain the significance of having radial symmetry for 2 marks. So radial symmetry means what? There's a central point that you can divide the living organism up into equal parts. And the significance of having radial symmetry would simply be it's going to allow the organism to sense food and danger from all directions. (3.1.6) has briefly described 3 characteristics present in the body plans of annelids that allows them to survive in their environment for 6 marks. So what does annelids have in their body plans that allows them to survive? Now there's a number of characteristics. We're going to go through it. Firstly, it has a hydrostatic skeleton which assists during movement. Secondly, it has a coelom which provides a circulatory system. It has a through gut which allows for more efficient absorption of nutrients and removal of waste. It has a high degree of cephalization, allowing for the concentration of sense organs and nervous to be controlled at the anterior end of the body. So any 3 of these characteristics along with the explanation would have been acceptable for this particular answer. Remember, we can change the word annelids and we can make it into chordata. We can also make it into arthropoda, because these are the more highly advanced animal phyla that we have studied. You could also have stated that there is a closed blood system which allows for faster transportation of blood within the blood vessels. The next question is a 3.2 that says read the extract below and answer the questions that follow. You will possibly have an extract in your paper as well. So our seed bank almost lost in serious war could help feed a warming planet. So Ali Shahada is a planet conservationist from Syria. His goal is to safeguard those seeds that may be hardly enough to feed us in the future, when many parts of the world could become as hot, arid, and inhospitable as it is in the Middle East. As the war in Syria intensified, Mr. Shahada was forced to leave the Encarta seed bank in Syria and flee his country. However, long before the war in 2008, ICARDA had begun to send seed samples to the Saville-built Global Seed Vault in Niue. This vault is built deep in the side of a mountain situated above the Arctic Circle. Mr. Shahada hunts for the genetic trait that was be the most useful in the future: resistance to pests and blistering winds and the ability to endure intensely hot summers. He tries to select for these traits and breeds them into the next generation of seeds. First question says, state 2 benefits of seed banks indicated in the above extract. So first benefit, it ensures food security. Secondly, the preservation of seeds that are resistant to pests. You could also see the preservation of seeds that are resistant to harsh weather conditions such as heat, drought and wind, because that was mentioned here in the paragraph. 3.2.2 explain why seeds originating in the Middle East would be suitable for cultivating in all parts of the world in the future. So these seeds are adapted for survival in hot and dry conditions that may occur in all parts of the world in the future. 3.2.3 state why the location of the Saville Global Seed Vault may be regarded as ideal for seed preservation. So you'd have seen that they would have spoken about cool conditions in Arctic polar regions, as well as a secure location in the deep inside the mountain. You'd have seen it stated here in the paragraph. The last question, 3.3, says describe the process of reproduction in angiosperms for 7 marks. Please make sure you are able to describe reproduction in angiosperms, gymnosperms, bryophyta, and pteridophyta, because this will possibly be asked of you. It's a possible longer question. So let's go through it. Firstly, the male reproductive structure of the angiosperm is referred to as the stamen and comprises of the anther and the filament. Female reproductive structure is referred to as the pistil and comprises of the stigma, style, and ovary. So when pollen grains are ripe, they are transferred using a pollinating agent, which will either be wind insects or birds. They're going to be transferred from the anther to the stigma. Pollen is going to move down the style to the ovary, and the pollen grain is going to comprise of the male gametes, which then reaches the ovum, which is the female gametes. It fuses to form a diploid zygote. So the pollen grains contains male gametes, which fuses with the ovum to form a diploid zygote. The zygote forms an embryo to form a seed. The seed is then enclosed inside a fruit. And when the fruit then eventually breaks off to start the process all over. Please make sure you're memorizing these points. So any 7 of these points would have been acceptable for 7 marks. That brings us to the end of the section where we looked at section B, questions taken from a previous control test to prepare you for your term 1 control test. Please make sure that you are studying all 3 topics: microorganisms, plant diversity, and animal diversity, and expose yourself to as many questions as you possibly can from previous control tests, as it will really assist you in devising and doing the best you possibly can. I wish you all the best for your upcoming March control test, and please be calm and contented and go into that control test with a positive frame of mind. I wish you all the best for your upcoming control test. Thank you very much for taking the time to watch this video.